Does the flagella of Vibrio correlate with virulence? By James, Alfredo, Jeff, and Josh. Biol 240, General Microbiology. Professor Chris Smith, PhD. April 28, 2025. Alright, microbiology friends, let's talk about a bacteria that loves the beach more than we do, Vibrio. It's a gram-negative, comma-shaped little swimmer that thrives in warm, salty, or brackish waters, where rivers flirt with the ocean. Now, don't let its love for tropical vibes fool you. Some Vibrio species cause serious trouble like cholera, aka severely watery diarrhea, and wound infections that can become dangerous fast. You'll often find Vibrio in raw and undercooked seafood, especially oysters, so slurp at your own risk. So while Vibrio might seem like just another beach bum microbe, don't be fooled. It's got a mean streak. Whether it's hitchhiking a ride on a raw oyster or sneaking in through a tiny cut, it knows how to make waves in the worst way. So yeah, enjoy the beach, but maybe don't invite Vibrio to dinner. Think of Vibrio's flagella like a high-performance outboard motor made of the protein flagellin. These long whip-like structures stick out from the bacterial body and spin like tiny propellers, launching the bacteria through liquids like microscopic jet skis. No arms, no legs, just pure spin power. But flagella aren't just spinning for style points. They're guided by chemotaxis, meaning Vibrio can sniff out the good stuff like nutrients and steer clear of danger, like toxins. They're like hangry toddlers with GPS, headed straight for the snacks and away from trouble.
Our hypothesis is, do generic variations in Vibrio flagellar genes correlate with increased virulence? If true, then species of highly pathogenic Vibrio would have similar sequences of flagella. There would almost be no similarities between highly pathogenic Vibrio flagella sequences to rarely pathogenic Vibrio flagella sequences.
For this presentation, we introduced two important Vibrio pathogens, Vibrio bolificus and Vibrio cholera. Both are motile flagellated bacteria and both cause serious diseases in humans. But what if the key to their pathogenicity lies in their flagella? Let's explore each of it. Vibrio bolificus is found in warm coastal waters. It causes wound infections that can rapidly escalate into necrotizing fasciitis, sometimes called flesh-eating disease. It's among the most lethal non-cholera Vibrio, especially dangerous to immunocompromised people. Vibrio cholera causes cholera, a waterborne disease characterized by extreme fluid loss through diarrhea. It thrives in contaminated water, often after natural disasters or in areas lacking sanitation. Though it is spread differently, it is also highly pathogenic. How? We learned in class that pathogenic strains of Vibrio cholera result from horizontal gene transfer by specialized transduction. Despite causing very different diseases, both species rely on flagella for movement, colonization, and possibly invasion. Vibrio cholera has a single polar flagellum. Vibrio bolificus can also express lateral flagella depending on its environment, possibly tied to its aggressive spread in tissues. Earlier, we saw both bacteria under electron microscopy. These physical structures, especially the flagella, may hold evolutionary clues about how virulence is encoded and shared. So to revisit our hypothesis, if flagella genes influence virulence, then highly pathogenic Vibrios should share similar flagellar sequences and differ significantly from those in rarely pathogenic strains. Next, we will explore a bit deeper into the genomes and also validate or invalidate our hypothesis.
We chose to focus on one gene to test our hypothesis, FLGN. This gene codes for a flagella synthesis protein and had three traits that made it ideal for us to use. As a protein that is responsible for assembling the flagellum, significant changes to this gene would lead to morphological differences in the flagellum or possibly flagella defects. Gene length is strongly conserved for this gene. Every copy that we found sequences for were the exact same length of 426 base pairs. This implies a level of consistency across the entire phylogeny, and that significant changes to this would cause this gene to stop functioning. Finally was availability. We found 33 different sequences for this gene across different Vibrio species, along with hundreds of whole genome sequences examples to compare it to. These two charts are comparisons of the base pair sequence of this gene to the whole genome sequences of Vibrio. On the left is V. vulnificus and on the right is V. cholerae. As you can see from the percent identical column, the red arrows, there is very little variation of this gene within the same species. Since this gene is a little over 400 base pairs long, each base pair difference is approximately 1/4%. V. vulnificus has about a 2% difference, 8 base pairs at its most varied in sequenced DNAs. At the bottom of the vulnificus chart, you can see a sharp drop-off when the next closest match is a different species of Vibrio, V. natrinogens, at approximately 100 base pair differences. We have even stronger evidence of conservation in V. cholera, where the top 100 matches, which is kind of where a basic blast search ends, across different strains of V. cholera have a 99% match. Here is the phylogenetic tree we constructed using a sequenced FLGN gene. It strongly disproves our hypothesis. You can see how the V. vulnificus and V. cholera, despite being very pathogenic, have a great deal of distance between their two branches of the phylogenetic tree. We are also able to get some additional reinforcement of our methodology. There were two different strains of V. sinaloensis that had the FLGN gene sequenced, and they show up right next to each other on the chart. When you compare just the two pathogenic strains of Vibrio, this specific gene only has a 61% overlap, providing additional support for the invalidation of our hypothesis.
Some implications for research. Our group concluded that this isn't the best way to examine virulence, as we're only examining the specific flagellar gene compared to taking a look at the entire genome and looking at specific factors that really tie into virulence instead of just one specific gene. In conclusion, our hypothesis proved false. Genetic variations in Vibrio flagellar genes do not correlate with increased virulence. Species of highly pathogenic Vibrio do not have similar sequences of flagellar, and no similarities of highly pathogenic Vibrio flagella sequences to rarely pathogenic Vibrio flagella sequences. In other words, this is not a good method of determining virulence.